unfold.
Welcome to the Deep Dive, the place where we synthesize complex research, extract the core truths, and provide you with a shortcut to being truly well-informed.
And today, we are confronting one of the most persistent anxieties of modern life.
The fear of cognitive decline.
It really is. It's this shadow that's over the amazing achievement of human longevity, isn't it? We celebrate living longer, but that celebration is always tempered by this dread of an inevitable erosion.
The slow fade of memory, the loss of, executive function, the weakening of wit.
And for almost the entire 20th century, that dread was, well, it was completely justified by the dominant science.
That's the critical starting point, isn't it? For decades, a prevailing scientific orthodoxy, the one that really dictated medical practice, educational policy, even how we viewed retirement, it was rooted in this belief that the brain was a static machine.
A machine with a fixed, non-renewable lifespan.
Right. So once that explosive wiring phase of childhood was over, you were just on a fixed downward slope. You got a finite number of neurons, and the only change possible was loss.
Exactly. And this belief, it was more than just some academic theory. It was gospel. It meant that old folk aphorism, "You can't teach an old dog new tricks,"
Was basically a clinical diagnosis.
it was elevated to that status, yes. It told everyone that new skills, new learning, that was fundamentally reserved for the young, that neurons could die, and that while you could maybe maintain what you had, you certainly could never structurally improve it.
Well, the sources you brought us for this deep dive, they introduce a modern and I think revolutionary paradigm, what we can rightly call the biological hope.
And it is a complete dismantling of that old dogma. The modern consensus doesn't see the brain as this fragile, obsolescent piece of hardware.
So what is it then?
It's a dynamic living tissue. It possesses this remarkable capacity to structurally remodel itself, to grow new connections, to optimize existing circuits, and even, get this, to generate new cells right until the final breath.
And this capacity, this neuroplasticity, it's not just for kids.
That is the critical point. It is not a feature reserved only for childhood.
So we're not just talking about abstract potential here. We're going to unpack the concrete evidence. We have material that goes from the microscopic view of a single synapse all the way up to structural imaging of human adults who have actively forced their brains to grow.
Precisely. We're going to review the surprising research from the physical expansion of gray matter in the heads of London taxi drivers to the cellular mechanisms found in these incredible individuals we call superagers who just defy the typical aging curve.
And the mission here is to move beyond just the inspiration of it all.
Yes. It's to provide you, the listener, with a detailed user manual for triggering and controlling this capacity for lifelong neural growth.
That sounds like a radical shift in perspective. Moving from a fixed liability to a dynamic asset. Okay, let's unpack this. We have to start with the sheer intellectual weight that needed to be overcome first. To really understand the magnitude of this neuroplasticity revolution, we have to acknowledge the intellectual fortress it had to breach. I mean, the static brain theory wasn't proposed by some minor academic.
Not at all. It was established by a towering figure, really the founder of modern neuroscience, Santiago Ramón y Cajal, right at the turn of the 20th century.
So his word was basically law.
Cajal's work was absolutely essential. I mean, he was the person who first proved the neuron doctrine, the idea that the nervous system is made of individual, discrete cells, not just one continuous net.
And he did this with that Golgi staining technique, right? The one that produced those incredible drawings.
Those breathtaking drawings, exactly. He revealed this intricate, complex world of neural structures. His observations were meticulous, world-changing, and for the most part, completely correct.
But when he applied that meticulous observation to the adult central nervous system, he came up short on one key point, didn't he?
He did. He was looking for signs of cellular regeneration, the way your skin or your liver cells divide through mitosis to heal and replace themselves.
He was looking for that in the brain.
And in the adult CNS, he observed a stunning lack of cell division. He saw established structures, he saw atrophy, he saw death, but he saw nothing new growing. And so based on his observations, which were of course limited by the technology of the time, he concluded regeneration was impossible.
And he didn't just whisper this observation, he codified it. It became the Cajal Decree.
He did. In his 1928 treatise, he delivered what he himself called a harsh decree. It was a statement of profound scientific pessimism, he wrote. In the adult centers, the nerve paths are something fixed, ended, and immutable. Everything may die, nothing may be regenerated.
Wow, that one sentence.
That single sentence effectively shut down research into adult neurogenesis and plasticity for decades. Though it's worth noting, he did end the statement with a bit of a hopeful challenge. He suggested it was for the science of the future to change, if possible, this harsh decree.
So if his observation was incomplete, why did it hold such sway for so long? What were people seeing in clinics that made his decree seem absolutely irrefutable?
The clinical alignment was just perfect. In the first half of the 20th century, we had no tools to support brain regeneration. If a patient suffered a catastrophic brain injury, say from a stroke or a traumatic accident, they rarely, if ever, achieved full functional recovery.
So the lost motor control or language ability, that was largely permanent.
It was permanent. And at the same time, memory seemed to naturally falter with age. These clinical realities just reinforced the notion that the brain, unlike almost every other organ, was non-renewable. It was a fixed resource you just used up.
And that biological view, which was the consensus in neurology and medical schools, it then seeped into the broader culture. And it was massively reinforced by the rise of the computer.
Oh, the computer metaphor was so powerful and so limiting. The brain became the hardware.
Right. Childhood was the programming phase. You're installing the operating system, the core software.
And adulthood became the long execution phase, just running the code you already had.
And aging, that became the terrifying reality of hardware obsolescence.
Precisely. This created a profound intellectual and societal paralysis. We call it a use-up model of cognition.
What does it mean, a use-up model?
It means that since the hardware couldn't be repaired or upgraded, any intense mental effort in old age was viewed not just as futile, but potentially damaging. You were just wearing out the remaining good components faster.
That has devastating implications for educational policy, for lifelong learning.
It absolutely did. If the scientific assumption is zero biological return on investment, then education systems were, quite logically, designed almost exclusively for the young. And worse, the advice given to older adults was often to rest their minds.
Avoid challenge. Withdraw from the cognitive friction of novelty.
Exactly. Which leads us right back to that self-fulfilling prophecy you mentioned.
The dogma accelerated the very decline it feared.
Yes. By encouraging seniors to withdraw from the challenge and novelty that we now know are required to maintain neural health, society inadvertently accelerated synaptic pruning and atrophy. They basically guaranteed the outcome that 𝐏𝐃𝐂 had predicted, ignoring the most critical biological principle of all, use it or lose it.
So the dismantling of this massive 80-year-old scientific consensus, it was a slow siege. It was carried out by researchers who dared to question the foundation. And our sources highlight two pioneering figures, Michael Merzenich and Fred Gage. Let's start with Merzenich. He challenged the very geography of the brain.
Right. Merzenich, working in the 1970s at UCSF, challenged the core assumption that the cortical maps, the areas dedicated to specific sensory or motor inputs, were rigidly fixed by genetics in childhood.
So the old model said that the part of your brain responsible for, say, your index finger was set in stone, its location, its size, all of it.
Forever. Merzenich decided to test this with his famous micromapping experiments on owl monkeys.
Okay, so what was this setup, and what did the static model predict would happen when he messed with the input?
So he used very precise electrode mapping to visualize the somatosensory cortex. He figured out exactly where the signals from each finger landed. The experimental manipulation was simple, but radical. He severed the nerve that carried input from a specific finger.
Okay, so the static model would predict that the corresponding area of the cortex should have just gone silent.
A permanent, unrecoverable, dead pixel on the map. That's the prediction.
But the brain, as a biological system, it hates unused territory.
It absolutely refused to accept silence. The radical result was that within a matter of weeks, the cortical zones representing the neighboring fingers, they physically invaded and commandeered that silent zone. The neural real estate was ruthlessly repurposed.
That is the first concrete proof that the brain's geography isn't a fixed blueprint.
It's a dynamic, competitive economy, constantly vying for resources based on continuous sensory input.
That's astonishing. But he pushed it even further with the controversial blind ferret experiment. This one seems to argue that the tissue itself is almost interchangeable.
That's the idea of pluripotency, that the tissue of the cortex is largely general purpose. The experiment was wild. It involved surgically rewiring the optic nerves of developing ferrets. So instead of routing the visual signals to the visual cortex in the back of the brain,
they sent them somewhere else entirely.
They rerouted them to the auditory cortex in the temporal lobe, the area that evolved to process sound.
So the static model would predict just neurological chaos. The hearing cortex gets visual data, it has no way to process, and it basically crashes.
Instead, the result was a revolution. The neurons in the auditory cortex, which were evolved to process sound frequencies, began organizing themselves into structures typical of the visual cortex.
No way.
They formed orientation columns and spatial grids. The hearing brain functionally learned to see using visual input.
So the takeaway is that brain areas specialize based on the data they get, not just some rigid genetic instruction. The brain is like a vast floodplain and experiences the water, cutting new channels.
That's a great analogy. But Marsenic's work, as groundbreaking as it was, still dealt with existing neurons.
Right. It proved remapping, which is a huge leap. But the deepest taboo, the one set by 𝐏𝐃𝐂𝐀𝐒, was neurogenesis. Could the adult brain actually grow new cells?
And this is where Fred Rusty Gage at the Salk Institute steps in, back in the 1990s.
And his breakthrough relied on finding a chemical marker, something that could prove cell birth was actually happening.
Exactly. They needed a reliable way to trace new cell division. Gage used something called bromodioxyuridine, or BRDU.
BRD.
Think of it as a chemical tracer. It's a synthetic molecule that, if you introduce it to a cell, gets integrated directly into the DNA whenever that cell divides. So if you give an organism BRDU, any new cell that is born will essentially have this tracer baked into its DNA, lighting it up for researchers.
And the human confirmation, that was a landmark paper in 1998.
It was. They examined the post-mortem brains of cancer patients who, ironically, had been given BRDU as a diagnostic tool, and they found thousands of BRDU-marked cells, new cells, in the dentate gyrus of the hippocampus.
The brain's core memory center.
Right in the memory center. This proved that new neurons were being born deep into adulthood, even in elderly and sick patients. The brain was structurally trying to regenerate itself until the very end.
That is amazing. But the skepticism must have been fierce. I mean, critics could have just dismissed these as, I don't know, non-functional cells or some kind of biological mistake.
And they did at first. So Gage provided the proof of functionality. He used sophisticated retroviral tracing in mice, tagged these newborn cells with a glowing fluorescent protein, and just watched them over months. The evidence was irrefutable.
What did he see?
He saw that the new neurons didn't just sit there. They grew complex axons and dendritic trees. They were actively plugging themselves into the existing, mature neural circuitry.
And they weren't just decorative, they were actually working.
They were. Electrophysiological tests confirmed they were firing action potentials, the electrical signal that neurons use to communicate. They were fully functional, integrated components of the network. This just crushed the deepest taboo of neuroscience.
And Gage also established that critical link, moving beyond just proving it exists to understanding the trigger. And that trigger was physical movement.
That's right. He observed that mice who had access to running wheels produced dramatically more new neurons in the hippocampus than the sedentary mice. This was the first concrete, confirmed link between physical activity and physical brain growth. It showed that neurogenesis is an activity-dependent process, heavily regulated by a protein called BDNF.
Okay, so by the early 2000s, we have the foundational cellular proofs from animal models. Remapping happens, and new neurons are born. But the next essential question for you, for the listener, is, does this actually translate to meaningful, measurable, structural change in a healthy human adult who's engaged in a complex task?
We needed that real-world high-stakes test, and the streets of London, specifically the drivers of those iconic black cabs, provided the perfect natural experiment.
The knowledge. This training sounds almost intentionally designed to push the limits of human spatial memory.
It's arguably the highest-level cognitive stress test imposed on any professional group. To pass the Knowledge of London, candidates have to commit to memory 320 specific routes, covering 25,000 streets.
25,000.
And logging 20,000 landmarks, all within a 6-mile radius of Charing Cross. And it's not passive memorization. They have to be able to instantly calculate the most efficient, dynamic route between any 2 points in that zone.
The effort is punishing. 3 to 4 years of full-time study with a failure rate of something like 67 to 70%. These people are essentially forcing massive cognitive infrastructure growth.
They really are. And a neuroscientist named Eleanor Maguire recognized this as a perfect opportunity to visualize plasticity in action. So she scanned the brains of qualified taxi drivers and compared them to matched control groups using structural MRI.
And the image of the taxi driver's brain gave us the first clear structural photograph of human adult neuroplasticity in response to massive cognitive effort.
The finding was unambiguous. The taxi drivers had a significantly larger, posterior hippocampus compared to the control group. And the posterior region is the part that specializes in spatial representation and retrieval. It is literally the storehouse for their massive internal GPS map of London.
Now, the key evidence that this was caused by the job, and not just that people with big hippocampi become taxi drivers, was the dose-dependent correlation, wasn't it?
That is the irrefutable data point. McGuire found that the volume of that posterior hippocampus correlated positively with the number of years spent driving a taxi.
So a 20-year veteran
Had a measurably larger posterior hippocampus than a 3-year rookie. This proved that the cognitive effort, the relentless learning and dynamic navigation was the driving force behind the structural growth.
The brain was responding to demand by physically adding gray matter volume to the area that was needed most. But let's zero in on the consequence of that trade-off, because the study also revealed a crucial limitation, this finite economy of neural real estate.
This is such a vital lesson about specialization. McGuire noted a biological compromise. While the posterior hippocampus was larger and optimized for retrieving that long-term memory map, the anterior hippocampus was significantly smaller than average. And the anterior region is the one involved in encoding novel visuospatial information, the machinery you'd use to quickly learn a new city.
So if the brain is like a highly specialized smartphone, the taxi driver's brain optimized its operating system for the massive, stable map of London, the posterior part. But it had less working memory, less RAM, for quickly downloading and processing new, temporary apps or novel routes.
That's a perfect way to put it. The brain operates on a finite, competitive economy. By dedicating so much gray matter and energy to store that massive, stable map, it repurposed or shrank the machinery for rapid and novel spatial learning. It's specialized. The takeaway is profound. We build the brain we actively use.
And the final piece of proof was ruling out just mere mileage as the cause. They had to compare the dynamic navigators to a group that drove just as much, but without all the complexity.
That was the brilliance of using the London bus driver control group. Bus drivers spend the same amount of time in traffic, dealing with the same streets.
But they follow fixed, highly repetitive routes.
Exactly. Their job demands execution, not dynamic problem solving. And the structural MRI results showed that the bus drivers had normal-sized hippocampi, completely indistinguishable from the control group.
So that isolates the structural change directly to the cognitive effort of dynamic, complex spatial navigation. Not just the physical act of driving. The message is clear. Complexity triggers growth. Passive routine encourages atrophy. Okay, we've proven the brain changes structurally in humans. Now we need to look under the hood. The process of learning and memory consolidation, it's driven by specific chemical and structural changes that happen at the cellular level. Let's zoom right in on the physical roots of thought. The dendritic spines.
Okay, so if you view a neuron as a complex tree, the dendrites are the major branches. The dendritic spines are these tiny, dynamic, thorn-like protrusions on those branches, and they are the precise locations of the synapse, the point where two neurons communicate.
And these structures, they're highly responsive to what we do, to our experience.
Yeah.
Our sources detail what happens when we try to learn a new, complex movement or a concept.
Right. When you fire a novel sequence of neurons, say you're struggling to master a new piano piece, the dendrites extend these thin, highly mobile, finger-like projections called filopodia.
So they're literally reaching out.
They're literally waving about in the space between cells, actively searching for a signaling partner, a neighbor neuron that is firing at the exact same moment. It's a physical search for synchronization.
And if they find one?
If that pairing occurs, if the two neurons fire together repeatedly, they form a temporary initial synapse. Now, if that activity continues with practice and repetition, the connection physically thickens. It transforms that thin filopodia into a sturdy bulbous structure called a mushroom spine.
And that thickening is the key.
That physical thickening accommodates more neurotransmitter receptors, making the connection faster and more reliable. That structural fortification is the cellular basis of a lasting memory.
But plasticity isn't just about building. It's also about a ruthless, brutal process of tearing down for efficiency. Synaptic pruning. The brain has to clear out the underbrush.
That's right. The brain isn't interested in being large. It's interested in being efficient and fast. And you can think of the brain as a sprawling, overgrown forest. To build a high-speed, reliable highway system, you need dedicated workers to clear away all the weak little trails.
And that's the job of the brain's specialized immune cells, the glial cells.
Specifically, microglia and astrocytes. They are the forest rangers, constantly patrolling for weakness.
So how does the brain identify which synapses are weak or underused? How does it know what to cut?
It uses a chemical flagging system. Weak, inactive connections get tagged with proteins from what's called the complement cascade, notably C1q and C3.
So they're like little warning flags.
Think of them as bright, chemical do not enter signs. Once a synapse is tagged, the microglia recognize that protein flag and they engulf the spine. They literally eat the synapse. It's selective elimination.
So if you learned French in college but haven't spoken it in 20 years, those complex language pathways, because they're not being used, are actively targeted for destruction to free up resources for things you're using.
That's the core mechanism. Use it or lose it is a biological imperative, not just a nice saying. Pruning removes neural noise and inefficiency, leaving only the strong, established highways. And this process is absolutely necessary to achieve true mastery and high-speed processing because it cuts down on the cognitive background clutter.
Okay, so the master molecule governing all of this growth and survival, the instruction signal for whether to build, maintain, or destroy, that's brain-derived neurotrophic factor, or BDNF.
BDNF is essential. It truly acts as Miracle-Gro for the nervous system. When BDNF binds to receptors on a neuron, it triggers a cascade with 3 crucial effects for plasticity. First, it provides neuroprotection. It prevents the neuron from dying. Second, it stimulates synaptogenesis, the growth of new dendritic spines. And third, it increases the signal strength across existing connections. That's a mechanism called long-term potentiation. If you want plasticity, you have to have BDNF.
Since BDNF is the chemical command for growth, the obvious question for our listener is, how do we, the learners, trigger this flow of fertilizer?
BDNF is an activity-dependent protein, and the research is crystal clear. The single most potent, reliable trigger is aerobic exercise.
Not weightlifting.
Not resistance training. This is getting your heart rate up for a sustained period. We're talking about the famous runner's high.
So part of that feeling is actually a flood of BDNF.
A big part of it, yes. Studies show that aerobic activity can triple BDNF synthesis in the hippocampus. It's thought that the increased metabolic demand and blood flow signal to the brain that it needs to fortify its infrastructure to support all that energy expenditure. It is the closest thing we have found to a genuine, side effect-free, cognitive performance-enhancing drug.
And the second trigger relates directly back to the taxi driver study and the enemy of neuroplasticity, routine.
That's novelty. The brain releases BDNF when it encounters something new or unexpected. If you take a completely new route home or learn a complex new physical skill or immerse yourself in a new language, the novelty reward system is triggered. This bathes the cortex in BDNF, essentially chemically tagging that new sensory information, marking it as important and worthy of being structurally encoded. Routine is the antithesis of BDNF release.
Now let's talk about the physiological mechanism that makes a signal stick. Long-term potentiation, or LTP. This is where Hebb's law, neurons that fire together, wire together, manifests physically.
LTP is the lasting, persistent enhancement of synaptic transmission. When a signal crosses a synapse repeatedly and with high intensity, and that means you are focused, alert, and applying effort, the receiving neuron responds by physically inserting more AMPA receptors into its membrane.
It's like widening a small dirt road into a massive multi-lane highway. It allows information to rush through faster and more reliably in the future.
Exactly. That neural pathway is now permanently optimized for high-speed traffic. Now, here is the crucial nuance for the adult brain. While the machinery for LTP remains perfectly intact in the aging brain, the threshold required to trigger it is higher.
So the too late myth assumes the machinery is broken. But the reality, as our sources show, is that the machinery is fine. It just needs a stronger, more intense initial signal to get it going.
It demands more focus, more sustained attention, more repetition, and more emotional or chemical energy to reach that threshold where the spine decides, okay, this activity is important enough to commit permanent resources to. But once you hit that threshold, the physical growth is every bit as robust as in a younger brain. The biological capacity is lifelong.
The ultimate irrefutable validation of lifelong neuroplasticity is found in this remarkable small demographic known as Superagers. They were identified primarily by researchers at Northwestern University, and these are individuals aged 80 and above who maintain the memory and cognitive profiles of someone 20 or 30 years younger.
They are the living evidence that the trajectory of cognitive decline is not a fixed script. They are actively rewriting it. If we look at the data on structural aging, it's pretty stark. Normal aging involves a steady cortical thinning. You lose about 2.24% of your brain volume per year after age 60.
And the superagers defy this expectation by more than half.
They shrink at less than half the normal rate, only about 1.06% per year. Their brains are aging on a completely different timeline. When researchers measure the cortical thickness of these superagers, it is often structurally indistinguishable from a 50-year-old. They've built an incredibly robust buffer against the passage of time.
And autopsies of superagers have revealed a fascinating biological advantage tied to these highly specialized cells, the von Economo neurons.
Yes, the anterior cingulate cortex, or ACC, a region that's critical for attention, error detection, executive control, and that subjective sense of effort, is notably thick and active in superagers. And autopsies confirm a density of von Economo neurons, or VNs, that is 4 to 5 times higher than their typical octogenarian peers.
VENs are those large, spindle-shaped neurons, right? They're found primarily in species with high social and executive intelligence, humans, great apes, whales. Why are they so important?
They're often called the supercommunicators, or the fast lanes of the brain's highway system. They facilitate rapid, high-stakes communication, particularly related to complex attention, decision-making, and social and emotional processing. The high density of these neurons in superagers suggests their brains are optimized for high-level, complex attentional demands and constant interaction with a nuanced world.
Which connects directly to their lifestyle. But first, let's talk about cognitive resilience versus resistance. This is where it gets, I think, startlingly elegant. The concept of cognitive reserve.
This is perhaps the most empowering finding of this entire deep dive. Researchers found that some superagers, upon autopsy, actually possess the beta amyloid plaques and neurofibrillary tangles associated with Alzheimer's disease pathology in their brains.
So they had the signs of the disease.
The physical disease was present, yet they had never shown any clinical symptoms of dementia in life.
So the function wasn't impaired. How does the brain pull off that kind of biological miracle?
It's not a miracle, it's infrastructure. The theory is that their high level of lifelong cognitive engagement, their relentless application of neuroplasticity, built a neural network that was so robust and redundant that it created a highly effective detour system.
A system that could route the signals around the roadblocks caused by the damaged tissue.
Exactly. They built multiple strong alternative pathways for processing information. So when one pathway was blocked by a plaque or a tangle, the signal simply routed down another equally strong avenue. They demonstrated that the functional integrity of the brain can withstand significant physical damage because they invested in building massive redundancy. That is cognitive reserve.
So the goal shifts from trying to prevent aging to building a system that can withstand the inevitability of age-related pathology.
That's it.
And while genetics surely plays a role in who becomes a superager, the lifestyle analysis points directly to replicable behaviors.
The behaviors are striking because they reinforce everything we've just learned about BDNF and plasticity. Superagers pursue high-effort mental tasks. They are not content with easy crosswords or passive television. They actively seek learning new, difficult skills. They embrace the mental friction.
And their social profile is also key, linking back to those supercommunicator neurons.
They're often described as social butterflies. They actively maintain wide, complex social networks. And think about it. Navigating complex relationships, reading emotional cues, mediating conflict, tracking multiple social narratives is one of the most cognitively demanding tasks we perform.
So that constant high-level social processing stimulates those crucial VNs in the anterior cingulate cortex, reinforcing executive function.
Constantly. And we also see that their lifestyle is one of connection and enjoyment over rigid ascetic restriction.
Right, it's not all about just grinding away at puzzles.
Absolutely not. They often enjoy moderation social wine with friends, for example, emphasizing that high-quality social health, reduced psychological stress, and a fulfilling life are just as vital to neuroprotection. Isolation is a profound enemy of the superager mind.
Superagers provide the statistical proof, but these individual stories, they translate that cold data into the warmth of human possibility. These are individuals who just flat-out rejected the too-late narrative and serve as the final proof that plasticity is truly lifelong.
They are. These are individuals who undertook massive cognitive challenges in life stages where society generally encourages withdrawal. Let's take Priscilla Sishine in rural Kenya. She spent her life as a respected village midwife, but she never learned to read or write.
And she didn't just decide to learn. At age 90, she enrolled in the local primary school, sitting at the desk with her great-great-grandchildren. The image itself is powerful, but the cognitives act is what really matters to us here.
Think about the neurological difficulty. Language acquisition, taking abstract visual symbols, letters, and reliably decoding them into sound and then linking them to semantic meaning. That is a massive cognitive load typically reserved for the highly plastic developing brain. For an adult, especially an octogenarian, the sheer volume of new information should have been insurmountable.
Yet she succeeded. She learned to read the Bible and, critically, learned to write down the extensive herbal knowledge she'd accumulated over 65 years.
Her success proved that the core circuitry for fundamental cognitive tasks like language acquisition and complex symbolic decoding remains fully accessible in the 10th decade of life. The brain's capacity to create stable, complex new pathways was just waiting, provided she applied the necessary effort. Her mantra, "Education is your wealth," became a biological reality inside her own mind.
And moving from language acquisition to high-level executive function, we have the story of Dr. Paul Ciramoni, the Indian humanitarian who pursued academic rigor late in life.
Dr. Ciramoni was already accomplished, but after retirement, he decided to formalize his life's work in theology and social transformation. At age 90, he earned his PhD.
And as you mentioned, this was not some honorary degree. This demanded full, high-level academic rigor. He had to write 6 scholarly papers, pass 3 grueling written exams, and defend a full thesis before a critical board.
This is such a powerful demonstration of the resilience of the prefrontal cortex. Academic research requires sustained attention, synthesis of complex data across decades of literature, logical structuring, high-level abstract reasoning. These are precisely the functions most susceptible to age-related atrophy.
And Ciramoni's success proves that executive decline is not inevitable. The prefrontal cortex can remain incredibly sharp if it's consistently challenged by novel, sustained, complex demands.
Absolutely. And finally, a completely different type of challenge, focusing on the motor system, which is often perceived as degrading even faster than cognition. John Lowe, the British grandfather who took up classical ballet at age 80.
At 80.
Yet.
Ballet.
The physical demands of ballet are a severe neurological challenge. It requires extreme balance, constant proprioception body awareness, and the acquisition of a complex, novel motor memory. It relies heavily on the cerebellum and the motor cortex. These are the functions that can degrade rapidly with age, often leading to instability and falls.
Yet he applied the rigor we've been talking about. Daily practice, embracing the struggle. He even rigged a trapeze in his garage to help him.
He was literally demanding that his motor cortex become plastic again, and the result was astonishing. He performed professionally as a soloist at Ely Cathedral at age 88 and continued dancing into his 90s. He proved that the motor cortex and cerebellum can acquire complex new kinetic patterns, defying the perceived inevitability of severe sarcopenia and balance loss. The mind, when sufficiently challenged, maintains the body's complexity.
So the evidence is overwhelming. The brain can change. But we established that adult plasticity is not the default state, like it is in childhood. The adult brain is efficient and rigid by default. So to leverage this power, we need specific protocols, a user manual, to chemically gate the brain and lower that threshold for long-term potentiation.
That's the key synthesis from our sources. These protocols move beyond general advice, and they focus on controlling the precise chemical levers, the neurotransmitters and neuromodulators that drive attention, arousal, and consolidation.
Okay, let's start with step 1. Get alert, epinephrine. We can't simply learn passively while we're relaxed. The brain needs a chemical wake-up call to pay attention.
Right. Plasticity requires high alertness and arousal. You have to spike your adrenaline, epinephrine, because it acts as a global broadcast signal telling the entire system, this information is important, encode it now. The protocol suggests simple, accessible techniques you can use immediately before a learning bout.
Such as the physiological sigh?
Exactly. The physiological sigh 2 deep inhales through the nose, followed by one long, extended exhale through the mouth, is a rapid neurological trick to shift your autonomic nervous system out of a calm, passive state and into a state of heightened alertness without inducing stress.
And what else?
Alternatively, short, deliberate exposure to cold, like a 30-second cold shower, or measured caffeine intake, can rapidly elevate epinephrine and prepare the neural machinery for encoding.
Okay, so step 2, get focused. Acetylcholine. Alertness gives you the energy, but you need to aim that energy at the specific neural circuits you want to change.
You need to engage the chemical spotlight. When you stare intensely at a visual target for 30 to 60 seconds right before your learning task, you trigger the release of acetylcholine from a region deep in the brain called the nucleus basalis.
And acetylcholine is the attention spotlight.
It is the ultimate attention spotlight. It literally bathes the active neurons, marking them for change.
So if I'm learning the piano before I start, I should stare intensely at the sheet music or a spot on the keyboard for a minute.
Yes, you are telling your brain chemically, pay attention to the specific visual, auditory, and motor inputs coming right now. Acetylcholine strengthens the signal of the active neurons, dramatically lowering the threshold required for those connections to undergo LTP. Without this focus, the learning signal is diffused and plasticity is minimal.
Step 3. Embrace errors. Dopamine frustration. This is where it gets so elegant and counterintuitive to what many people think effective learning is. Frustration is the medicine.
Most people stop when they feel frustrated. They interpret it as a sign of failure or low aptitude. But frustration is actually the sign that the brain is engaged and ready to change. The feeling of frustration when you make a mistake signals to the system that the current neural pathway, the one you just executed, failed to achieve the intended outcome.
And that error causes a spike in the neurotransmitter dopamine.
Exactly, which paradoxically enhances attention and encoding of the next attempt.
So the brain is getting chemically tagged to say the path you just took was bad. We need to pay extreme attention to the subsequent corrected path.
Precisely. That's why the protocol advises aiming for a Goldilocks zone failure rate of approximately 15%.
15%.
Right. If you are getting everything right, 0% error, you are just executing a routine. That generates no BDNF and no strong error signal. If you're failing 50% of the time, you'll quit, and the attempt is just too noisy. That 15% error rate is the sweet spot of mental friction. Just enough effort and frustration to trigger the dopamine-driven need to rewire.
Step 4. The gap effect. Microrests.
Yeah.
This is vital for encoding data during the practice session, not just after. The brain needs those buffer moments.
It does. Continuous practice without breaks is far less efficient for adult plasticity. The protocol advises stopping for just 10 to 60 seconds every few minutes during the intensive learning bout and doing absolutely nothing.
No checking your phone, no reviewing your notes.
Sort of quiet.
What's happening biologically during that short moment of stillness.
Research shows that during these micro-rests, the hippocampus rapidly replays the neural sequence you just practiced, but at speeds up to 10 times faster than real time. It's like hitting a high-speed rewind button. This gap effect massively accelerates the internal encoding and consolidation process, letting the brain quickly solidify the fragile new pathways before they're overwritten by new information.
And finally, step 5, deep rest, consolidation. The physical structural growth, the building of those mushroom spines, it doesn't happen while we are awake and struggling.
No, the actual physical rewiring, the structural changes to the spine morphology, occurs almost exclusively during slow-wave sleep, the deepest phase of non-REM sleep. The brain actively reviews and solidifies the day's learning, performing that critical pruning and growing. Prioritizing high-quality sleep after a focused learning session is non-negotiable for lasting plasticity.
And for those times when a full night's sleep isn't possible, our sources point to an immediate alternative.
Yes, using a 20-minute non-sleep deep rest, or NSDR, or a yoga nidra session, immediately after a focused learning bout has been shown to facilitate the structural consolidation. It acts as a powerful buffer for encoding before a subsequent sleep period. The core idea remains: the hard work is done during effort. The physical change is done during rest.
Let's pivot back to the selection of the task, because that is the most frequent mistake people make when they're seeking brain fitness. Our sources make a strong case for why routine mental tasks fail.
Right. Doing the same crossword puzzle every day or playing a familiar game of Sudoku, it just utilizes existing, strong, efficient pathways. It's execution. It doesn't trigger the BDNF cascade or require the focus and error signaling necessary for growth because it lacks novelty.
To truly optimize the brain, you must actively seek novelty.
You have to do things you are genuinely bad at, where your error rate is initially around that 15% mark. The mental struggle is the specific medicine required for neuroplasticity.
And of all the potential activities that combine novelty, effort, and sensory integration, the sources identify one intervention as arguably the most potent driver of lifelong neuroplasticity.
That is musical training. Learning a musical instrument, especially in adulthood, forces profound, simultaneous, multisensory integration. You are integrating vision reading notes audition, listening for pitch and rhythm complex motor control, using both hands independently, and emotional interpretation all at once. This holistic activity forces the entire brain to fire together in novel ways.
And the structural changes are a clear indicator of this full brain integration.
They're dramatic. Musicians consistently develop a thicker corpus callosum, that massive bundle of nerve fibers connecting the left and right hemispheres. This thicker bridge facilitates faster, more robust communication between the hemispheres, which improves generalized skills like memory, attention switching, and executive function.
And crucially, music provides those true transfer effects. It improves generalized abilities, unlike those single-purpose brain training apps, making it a supreme tool for building cognitive reserve. It really is. #tag outro.
So our journey today has taken us from the 𝐏𝐻𝐃 decree, which painted the brain as this fixed dying machine, to the biological hope, proving it is a dynamic, resilient garden capable of structural remodeling until the final breath.
The evidence is just conclusive and repeatable. Neurogenesis happens, cortical maps rewrite themselves based on input, and gray matter volume expands with sustained, complex effort.
This scientific shift really demands a matching philosophical shift in how we view the second half of life.
It does. The brain is fundamentally an organ of adaptation. If you feed it passive routine, it will become efficient at routine and will atrophy its capacity for learning. If you feed it novelty, complexity, and mental friction, it responds by building robust, redundant infrastructure. It strengthens the very systems that withstand the challenges of aging.
So the fear of cognitive decline is rational, but the historical default response of withdrawal and rest is biologically counterproductive. The scientific prescription is the exact opposite: engagement. Whether it is learning a new language like Priscilla Satine, engaging the executive function required for a PhD like Dr. Ceremony, or forcing complex motor skill acquisition like John Lowe, the method is the same. Do what is difficult and novel.
The biological capacity for growth doesn't lock at any specific age. The neurons are waiting, the synapses are ready to connect, and the BDNF is ready to flood the system.
The brain is, functionally and structurally, a muscle that grows until you die, but only if you choose to lift the weight required to trigger that adaptation.
And that leaves us with one final provocative thought for you, the learner. We know definitively that we can build a brain resilient enough to withstand the pathology of aging, a system so redundant it can route around roadblocks. So if cognitive optimization truly persists until the final breath, what complex new skill, the one you thought you were definitively too old for, are you now biologically required to start?